Hello all. Welcome to your lab 6. So this lab will be run asynchronously like normal. Any questions you have in this, trying to understand how to do the simulations, trying to understand exactly what's going on with this lab or how Hardy-Weinberg works, please direct to to Professor Sadigian, who's going to be your sub for this week. Okay. He's also going to be able to answer any questions about the lab PowerPoints and Lab 5 and the quiz and the exam, which is this weekend. Okay. So let's start. Something we have to know when we're talking about Hardy-Weinberg equilibrium, we talked about this last class. This Hardy-Weinberg is going to be on your exam, okay? An organism with two identical alleles for a character, it's homozygous for the gene controlling that character. Reasonable, right? An organism with two different alleles, it's heterozygous, and unlike homozygotes, heterozygotes are not true breeding. This is going to matter for the simulation that we are doing in lab, okay? So this just shows how that works, and definition of evolution is the changing of frequency of alleles. It's not just changing the phenotypes, how individuals look, but the idea is that the alleles, the frequency that they are presented is here, okay? So in the first generation, there's these characters. We have two homozygous recessives, one homozygous dominant, and one heterozygote. If we switch to the next generation, there being this, technically there's been no evolution. Because let's count. 1, 2, 3 big A's. 1, 2, 3 big A's. 1, 2, 3, 4, 5 little A's. 1, 2, 3, 4, 5 little A's. The population did not evolve because it has to do with gene pools and frequency of alleles. All right. Remember to be in equilibrium, not evolving. The following items are true. Random mating, everyone gets to mate. No natural selection, no genetic drift, no gene flow, no mutation in the big population. We're going to be mostly talking about the random mating, no natural selection, and a little bit in this class about these items here. Genetic drift, gene flow, and mutation. So something you need to be aware of is how P and Q work. Again, this is something that we worked through in lecture. Please watch the lecture. I also walked through it with you guys. But the idea is that P plus Q equals 1. These are the frequencies of the first and second allele. Generally, P is for the dominant allele. Generally, Q is for the recessive allele. And so these are the alleles. So when I counted up 1, 2, 3, 4, 5, 6, etc. for the big A or little a, I was referring to the frequency of the alleles. But you noted that there was a different number of homozygotes and heterozygotes. When you want to figure out the frequency of homozygotes and heterozygotes in a population, because there's two alleles in each individual, you have to square the whole thing, okay? So P squared is the frequency of the homozygous dominant, 2PQ is the frequency of the heterozygote, and a Q squared frequency of the homozygous recessive. So how do we get here? Okay, this is the quadratic equation expanded out. It's basically a, so if we imagine that we are making a Punnett square, a simple Punnett square, we build out that Punnett square, build, okay? But instead of putting big A, little a, we put the frequency that we would find a big A and the frequency that we would find a little a in the gene pool. Then we multiply them together using that AND rule, okay? The AND and OR rule. 0.7 times 0.7, well, that's 0.49, okay? And then we get 2PQ because in a, there's a frequency for getting the the heterozygote from the dominant from mom and recessive from dad, but then there's also one getting the recessive from mom and the dominant from dad. And so then there's the OR rule there, okay? the key thing to remember here is that if the population is done evolving and it is in equilibrium, then allele frequencies will not change over time, at least not significantly for a very large population. And again, you can look at it this way. Here's the gene pool. The likelihood that you're going to grab recessives versus dominants, there's a probability. And again, this is how you get to those numbers that we followed. Okay. And then you figure out how to find the frequency of the big A's. Big A's are going to be 49%, okay, and half of 42, because half of the big A little a's are big A's, okay, and then again 9%, and then half of that 0.42. You add those together, and you find Q and P. So let's follow how to solve some problems for Hardy-Weinberg. If you have genotypes for the organisms, you're going to figure out how many of each allele exists. Okay, again, if you have big D, there's 2 big D alleles per individual. This one has 1 big D and 1 little d allele per individual, 2 little d alleles per individual. You count the total numbers of the alleles, and you divide D or little d by the total number of alleles to get frequency. Okay, so that's how you get those percentages. If you have like 100 here and 200 here, then total you have 300 alleles, and then you divide 100 by 300 to get your number, your percentage of one type. Okay, frequency just indicates it's sort of percentage, but it's in the decimal form. To figure out based on recessives, okay, if you have the recessive individual frequency, this particularly is for ones where you can't tell the difference phenotypically from a individual who is homozygous dominant than an individual who is homozygous, who is a heterozygote. You can tell who the little a individuals are, right? The idea is that you can tell who they are by Q squared. You know how many people, so Q squared is the frequency of the homozygous recessive. They are the only ones who are going to express the recessive trait. So if you look at it at a population and you have, 2,000 individuals with brown eyes and 500 with blue, you know that Q squared is 500. Take the, then you find out 500 divided by 1,500, that's the frequency, okay? Get your frequency. So if you have 16 individuals, 4 are homozygous recessive, then Q squared equals 4 out of 16, or 0.25. To get Q, you take the square root of Q squared, which is 0.5. Remember, important, when you have the frequency of individuals, you do not have Q, you have Q squared. Remember to first find the frequency of the homozygous recessive, and then take the square root, okay? If you have Q, you have P. 1 minus Q is 0.5, and that's P, okay? Plug those in, and you're going to get any genotype frequency. If you have 2 of the genotypes, well, you don't have to do the third genotype, because you can just add them up, and they will all add up to 1, okay? Okay, remember when you're solving problems for this, if you have P, you have Q, vice versa. If you have frequency of recessives, you do not have Q yet, you have Q squared, recessive individuals. Make sure you're finding the correct question, and if you want to find P, you can't use Q squared to find P. You've got to get Q first, okay? This is because of the difference between frequency of individuals and frequency of alleles. Some of the alleles are hidden, okay? Hidden recessives in this 2PQ part. Q hides behind the P, which is generally dominant. And so you have all these individuals that look dominant but are actually recessive, have recessives in them, okay? If you're asked to find the frequency of a genotype, you have to find P and Q and then plug into the correct part of the equation. You have to have P and Q to do any of those. If I ask you the frequency of the homozygous recessive and you have Q squared, then you can do that, but you have to find P and Q to get P squared or Q squared, If you're asked to find a number of individuals in a phenotype, okay, let's say I ask you, well, how many of these brown-eyed individuals are heterozygotes, or how many of these individuals, according to Hardy-Weinberg, if I give you P and Q, how many would be expected to be brown-eyed? Then you'd find frequency for each genotype that looks like that phenotype, add them together, multiply the total population, and get the frequency, okay? But again, you have to find the frequency of the genotypes that add up to that. All right, let's go to simutext, okay? This is going to be the major part of it. I just wanted you to help hone in on solving problems, and I'll work through some of the problems with you guys, okay? All right, here's my simutext. Okay. We're going to switch around for a minute because I realize I need, oh, don't mind that for a moment. I'm going to change the display. No, wait. Give me 1 second, realizing I'm going to pause this. All right, I figured it out. I was very confused. I wanted to understand what to do, and now I have figured it out. We are back to doing simutext. Okay, let's start from the beginning. Sickle cell disease and malaria. This is where we're going, okay? So this talks about sickle cell disease, okay? Go through this, read it clearly, but we've already talked about the fact that sickle cell is a disease that is more common in in sub-Saharan Africa, in large part because it provides some immunity to malaria. All right, so this is a labeling activity, okay? So the idea is that we already talked a little bit here, but we have the chromosomes here. These are homologous chromosomes, chromosome from mother and father, okay? This is pointing to this little a, that's an allele. This is pointing to this little b, that's an allele. And these are the locuses, homozygous genotype. It's big A, heterozygous genotype, it's big B, little b, okay? This is just basic stuff. And the basics of how sickle cell works, okay? And maybe genetic disease could be beneficial, okay? Obviously, people who have 2 copies of the sickle cell disease allele have the disease and have some issues there, but it's a codominant trait, which means that heterozygotes will have a slightly different phenotype, and maybe we can see why they might be at an advantage, okay? So, make this big, as big as I can get it at least. Okay. Here's the deal. This lab investigates the connection between malaria and sickle cell disease with a simulation that tracks allele and genotype frequencies. Allele and genotype frequencies, we're talking about Hardy-Weinberg, remember. This is going to build up to it, but this is a piece, okay? So, look for the simulation control buttons, they're down here. Click the go button, okay? And we're going to run this. All right, so let's see the individuals here, okay? We have, this started out as, so I want to pause for a second, okay? Stop for a moment. We see that it started out as the normal allele being 75% of the individuals, and only less than 25% was had sickle celled. And what you see here is the red individuals being sickle cell individuals, the blue being individuals who are normal in that they have all normal blood cells, and then the heterozygotes are purple. You look at that, wow, that's a lot of heterozygotes. What you can see up here is the mosquitoes that live in this area. And we have a malarial death rate of 20%. That's a lot. Malarial death rate, though, is only affecting those who are maybe a certain genotype, okay? This talks about selection in a specific environment. What we have here is that instead of it going to be 75 and 25, it starts to be very even, 50%. Remember we talked about stabilizing, balancing selection? Well, this is balancing right here, okay? It's balancing the difference between the two because the heterozygotes are more common in this area, okay? So let's go. We are now with the deaths here, okay? Past 100 generations, okay? We're right at about 50% of each allele, and it quickly changed because of these deaths. So let's look. After 100 generations have passed, which is the following statement is true. Most people are susceptible to malaria because they have this big A genotype. And the answer is true, okay? Most people have sickle cell because they have big S. Sure, but the concept here, though, is that the malaria is hitting mostly these big A individuals. But another big piece here. Well, the most people who have the big A, and most people who have the big S are there, most of the people are purple. Most people have neither disease, which indicates that there is actually a benefit to being a heterozygote, right? Most people now in this population, all those purples, are big A, big S. They're not susceptible to malaria, nor will they have sickle cell disease, okay? So, what happens to the frequency of the S allele? It increased, look at that, okay? We want to see it rapidly increase, okay? Rapidly increase to about 0.5, then remains constant. And because each person has 2 alleles at every locus, the frequency of an allele in a population of size n is the number of copies of the allele divided by 2n, which is what we did with solving some of those problems, okay? There's 250 HBA alleles in a population of 500 people. What's the frequency? Well, we know this, right? I just went through how to find the frequency of different alleles. We have a population of 500 people. Each of these 500 people have 2 alleles each. If there's 250 alleles total, it's actually 1,000 alleles in 500 people. The frequency is going to be 0.25, right? Do you see how I did that? Remember, HBA and HBS, those are the only alleles in the population. So if there's 500 people, and they each have 2 alleles, then we have 1,000 alleles. 250A, that's 25%. Okay? Again, it shows you how I built it out here. All right, so answer that based on that. Okay, I'm not going to answer this one, but do the same idea. 500 people in a population. Remember, 500 people equals 1,000 alleles. 150 are homozygous, HBA, 150 are this, 200 are this. Remember how I built that out, right? Let's go back real quick to the PowerPoint. Okay? That's solving problems, right? To count them, 2 of the first allele per individual. For the homozygous dominant, 1 and then 2, okay? Remember how we counted them before. Okay, let's go back. We take the A1, okay, let's say HBA, and we take the homozygous dominant, and we multiply that by 2. So if we have the individuals, if there's 150 who have big A, or the HBA, all right, then there is going to be 300 total of these in that genotype, and then we just take the straight number, okay, if there's 150 of the others. So let's walk through this, okay? So 500 people in population, 1,000 alleles total. but we have 150 are homozygous HBA, okay? It means 300 alleles in there are this big A. Remember, because that's the big A one, okay? 150 are the HBS. Okay, that's the big S, so it's the other homozygote. 200, so what we actually are going to have is 300 here, plus we want to let's just find the HBA, okay? 300 in these 150 homozygotes, and 200 in these heterozygotes, because only half of them, so you don't multiply by 2. Basically, there's as many alleles as there are a number of people, okay? 500 total. It's going to be a 0.5 situation, right? But that does make sense. Build it out, okay? It's going to tell you whether you're wrong and show you how to build it. Okay. Without resetting the simulation, slide the malaria death rate slider all the way to the left. Whoa. It's no longer, you're outside of the realm of malaria. It's going to pop up in the lecture that you were getting today as well, okay? Right. Let's go. And we're going to make it stabilize. No more up and downs, okay? Go. Whoa, suddenly, what's going down? HBS. No more mosquitoes in the sky. There's some heterozygotes. But sickle cell is so detrimental that in an area without malaria, there is no evolutionary reason to keep that. Basically, the people with sickle cell are being selected out because there's no selection to keep the heterozygotes. So the heterozygotes, while they're still having homozygote dominant, homozygous recessives, if they mate with each other, in that there tends to be more homozygous normal allele, and that starts to dilute away because the sickle cell individuals are not surviving well either. When you eliminate malaria from the population, malaria deaths ceased. What happened to the allele frequency? We see this right here, okay? Went away. Frequency HBS goes to 0. No one's dying of malaria or sickle cell, okay? So it confers no advantage. It eventually disappears or becomes extremely rare. Remember, white people whose ancestors are in Europe occasionally have the sickle cell allele because it is a minor mutation that can pop up in any population, but it's going to be extremely rare versus being pretty common. All right. Where should it occur and why? It's because where mosquitoes are, mosquitoes love standing water. And so we have low precipitation levels, here we have, and think about where the mosquitoes live, etc. And the precipitation, why would it follow that? Well, this is the ecology of the area. Is that in areas with more precipitation, you have more mosquitoes, you have more, etc. Okay? All right. So now we're going to come out to, again, it's a very complex disease. We are going to click the button for different moisture and mosquito levels, and you should see the malaria prevalence, okay? So let's click here. Okay, see what the pattern is there. Click here instead. Some mosquitoes slightly wet. Oh, see, stops being as beneficial. Dry, no mosquitoes. And you see that going away, okay? Malaria prevalence the highest. So I want you guys to run through this, okay? Build this out. Follow this here, okay? So you're gonna click step 100 to just run it for 100 generations, okay? Doop. Step 100. just wait for 100, okay? I don't know why mine's not working. All right, there, it stopped at 100. Ta-da! This is the dry region, so go ahead and write down the frequency of the HBS and the sickle cell deaths. Oh, there's none, okay? So walk through, fill it all out. You guys do have to fill them all out and actually run each one of these simulations and get the numbers, okay? And then describe the patterns. But that does make sense, right? Where malaria is common, people with which genotype are the most fit? This should be clear to you, okay? And in which regions would the HbA/HbS genotype be associated with an overall fitness disadvantage, and why? Fitness disadvantage means that their children are also fit. Remember that individuals who have heterozygous can create both people with a normal genotype and sickle cell. So think about that, okay? All right. We talk about fitness and natural selection, the variation, okay? Differential fitness, variation, heritability, differential fitness, okay? And then we talk about the conditions. So this is all natural selection pieces that we've talked about and other pieces, okay? All right. So I'm going to skip ahead again, which is the following: evidence that malaria creates a heterozygous advantage at the HB locus. Now we have this piece that they're talking about. Read through them. You should be able to figure that out, okay? Again, these are questions that you can answer wrong and it doesn't matter. Just figure out why it's right or wrong, okay? Let's move up. Okay, then test your understanding real quick. Very wet region, lots of mosquitoes. Entire population escaped them by moving to an unpopulated dry region. How would that change people's fitness? Okay, so think about that. Relative fitness of which one would do what, and then what would happen, okay? Think about those clearly. There's going to be several different parts to that. Okay, next. How do new alleles actually establish? Okay, so this talks about random mutations. This is something that's brought up in class by your substitute professor. Okay, how do alleles come about from random mutations? Okay, some communities are quite remote, infrequently receive immigrants. Given what you know about the heterozygous advantage, if an immigrant with the HBS allele appeared in an isolated African population where the HBS allele did not already occur, could you predict whether the allele would persist? Okay, so this is talking about regions here, okay? All right, so this is a similar pattern. You're going to be doing simulations. Initial numbers of HBS carriers to 1, okay? This is assuming this person is going to mate with the population that doesn't have sickle cell, and then that will persist. How long would it take? Okay? What happened to it over time in the different areas? Would it persist in various areas? So just run through them, okay? Trial 1, trial 2, trial 3, okay? And then see those patterns and see if natural selection actually impacts them, okay? All right. What is persistence? So this is talking about persistence. Which hypothesis is best supported by our data? Okay, so all of these are here, and it talks about genetic drift and the initial frequency for different populations. Obviously, genetic drift sometimes varies here. I'm going to move forward, okay? I'm going to get, again, you're going to have to work on getting that frequency there. But I want to get to the Hardy-Weinberg principle, okay? So, we want to know, is evolution stable in the areas where the mosquitoes are persisting, okay? So we're going to, how do we tell if evolution is happening if we can't see the actual circumstances causing it? Is it happening? Well, we need to find out if alleles are changing. And that's the Hardy-Weinberg. That's the part that I went through in more detail in my presentation, okay? So I'm going to move forward here. All right, this talks about all this deriving here. But when we talk about genes of interest, we're talking about the, so p squared for this one is going to be the normal allele versus the recessive, the disease allele, okay? So here we go. HBA, P times P squared, okay? Q squared is going to be S, and then the 2PQ is going to be the heterozygote. And the probability that an individual will be homozygous is the same as P squared plus Q squared, okay? Not a heterozygote. See? All right. Then it walks you through problems. Now these problems, again, can be a little bit confusing, but this is where some of the patterns that I talked about in the presentation come up, okay? So I want everyone to be focusing on exactly how you build these. It's going to help you build them up, okay? If the frequency of the big A, if P squared, this is what this is asking you. If P squared is 0.1, what's the value of P squared? It's the same. Oh, wait. Oh. Frequency of HBA homozygotes is 0.1. What's the value of P squared? Okay. Oh, 0.1, sorry. There you go. I was like, it's the same thing. No, I put the wrong number there. My fault. Okay, suppose the frequency of the homozygous individuals is 0.01. Frequency of the heterozygous individuals is 0.18. And then what proportion should be this one? Remember, everything adds up to 1. So just take this 1 minus this, and then you have your homozygous S. Use a calculator. So it's select calculator from the module contents, okay? Here's your calculator. Or, you know what you can do is, this just opens a regular calculator on your computer. You can use your phone. Don't worry about it. But in this way, I want you guys to figure this out, okay? Build that out. Remember, P squared, HBA individuals, 2PQ, HBA HBS individuals, plus Q squared equals 1. So figure out what these two are added together and then figure out what you need left to get to 1. You guys can figure this out on your own, okay? It's the frequency of individuals who are homozygous, HBS 64, and the population is a Hardy-Weinberg equilibrium. What's the expected frequency of this allele? This shows you. We have 0.64. That's Q squared. Take the square root of 0.64. 0.8. 0.8 is Q. P is 0.2. So the HBS allele, remember, if you have homozygous recessive individuals, you don't have the frequency of the S allele yet. Remember, heterozygotes have some of those in them. So it's actually going to be, again, 0.64, square root of 0.64. And it shows you how to do that, okay? So take P squared here, take the square root of that, 0.59, and then you have Q and P, okay? You can do it if you have P, you have Q, if you have Q, you have P. So we're going to find Q here. So let's just type that in, 0.8, okay? Because 64, square root of 64 is 0.8. There you go. See? And then you have this. It's a frequency of this allele if Q is 0.1. What is the expected frequency of these heterozygotes? 2PQ. 0.1, then HBA is 0.9, so 2 times 0.1 times 0.9, okay? 2 times, so 0.1 times 0.9 is 0.09 times 2 is 0.18. Okay? Hardy and Weinberg would be proud if you figured that out. All right. And this is where you start to play with them again. Here we have your handy dandy Hardy Weinberg calculator. All right, frequency of the first allele, and it's going to give you all the expected values. Oh, well, how handy dandy, okay? Figure out which statements below are true. Remember what I said. Things have to stay the same between generations. It has to conform to all of the requirements. And remember how that works. Okay, what is what? All right. So let's, according to calculate what's the expected frequency of the heterozygotes when p squared is 0.1, all you have to do is say p is 0.1. Oh, look at that. Q is 0.9. Q squared is 0.81. P squared is 0.01. 2pq is 0.18. All those things we figured out already, okay? Play with the slider here, okay? Hardy-Weinberg is what's called a null hypothesis. It's a null model. If no evolution is happening, then all of the expectations of the Hardy-Weinberg equilibrium will be met. Okay? So, in the populations that you and your rivals are studying, which hypothesis would be supported by observed genotype frequencies that are close to those predicted? If remember, this was the similar thing that we went through with that MN, that we expected it to be in equilibrium, and we found it out by taking the genotypes, figuring out P and Q, and then saying, does that match the expected? And we found it did, so there was not evolution happening on that. Okay? Null hypothesis. So in these populations, if there is, okay. All right. Research team racing against another research team for the first evidence for or against a heterozygote advantage for HbA, HbS individuals. Okay? Hardy-Weinberg is coming to the rescue. The null hypothesis is stating that there is a heterozygote advantage because, so it'd be an alternative hypothesis. The null hypothesis says there shouldn't be a heterozygote advantage. For there to be evolution happening, we have to say an alternate hypothesis, okay? Oh, sorry. Heterozygote advantage should lead to, okay, so if we found them close to predicted, which again, we're not finding, sorry. My brain went ahead of me here. The null hypothesis is going to say that everything's the same. There's no advantage for anything. There's no selection. Okay? So that. The alternative hypothesis is indicating what we saw before, that the HbA, HbS genotype will occur more frequently than predicted. Okay? So, now you're going to run the models. Okay? Go through. This is going to tell you, you're going to select a very wet. We know there's lots of mosquitoes there. Step 100, allele frequencies tab of the graph. Okay, so we're going to watch HbA and HbS. We're going to look at the observed HbA frequency at generation 100. And then we're going to check out the genotype frequencies. All right. And then we're going to see expected versus observed. This is literally a chi-squared pile. I don't think they're going to make you do the statistics, but this is the possibilities here, okay? All right. So let's start with this. I'm going to help you run through it, okay? Just this part, because I believe there's more parts here. You just keep going through that, okay? So let's go back. Generation 100, we're going to step 100, okay? It's going to stop at 100. Okay, what is my HbA frequency? Doot. 0.43, okay? Now we're going to go to genotype frequencies, and whoa, we see a lot of these, okay? But let's go back here. Allele frequencies here. When we talk about that, one of the key things you want to remember is that we would expect 2pq, right? So let's calculate that out before we go anywhere, before we even start. what would 2pq be, our expected heterozygote frequency? This would be 2 times 0.43 times 0.57. 2 times 0.43 times 0.57. 0.49. We'd expect it to be about close to 50%. If there is no advantage, if everything is in equilibrium and there's no heterozygote advantage, this is what we expect. That is above 0.59, not 0.49, 10% higher than you'd expect. And these are lower than you'd expect, okay? We've observed, and we are going to do expected frequencies, okay? The Hardy-Weinberg calculator, you can do this, play around with it. You don't have to do it on your phone, but you can see that you'd expect them. And now it's going to show you that what's the difference literally between your observed and expected frequencies of heterozygotes. That is, we go here, is 0.1, okay? 10% difference, okay? Difference in the frequency of more than 0.05 is statistically significant. We have significance. Ta-da! The alternative hypothesis is supported. This has you go through and keep going, slightly wet, and the dry region, etc. It goes, and then you're looking at the different death rates and genotype frequencies in the dry region with no sickle cell. And then on your own, you're going to explore a little bit more, look at these different things. There's complex models and such. You play with the parameters, see how that works, because you're going to have to be able to predict and describe later, okay? All right, you can keep playing it. It's still available, and then you have your graded questions, okay? And everyone's going to have different sets of graded questions. This helps you understand, right? That if it was in Hardy-Weinberg equilibrium, there would be no selection and no preference for any genotype. Instead, what you're seeing is preference. And so you're going to calculate out how that works and also solve some of the problems that it presents to you. I will present you problems in lab quizzes 3 and the lab exam that have to do with, if you know Q squared, what is P squared? If you know, there's a variety of different things, okay? So you're going to be able, what is P, what is Q? What does it mean if the genotype frequencies don't match? It means that one of them's doing better than another, etc. Okay, so if something's higher, then selection is happening to keep it high. That's all I have. Have fun playing the games and doing all the pieces, and have a good week. And I will see you back the week after, October 17th, I think. 16th. That 2nd October day. All right. Everyone have a great day.